Gene regulation is the mechanisms and systems that controls the expression of genes. Constitutive genes are continuously transcribed and therefore always expressed and not regulated. Bacteria use regulatory genes to control gene expression. Regulatory genes have their own promoters. These genes are transcribed into strands of mRNA and translated to regulatory proteins. These proteins bind to the DNA strand and help control expression of structural genes by increasing or decreasing transcription. Operons control transcription in bacterial cells. An operon is a group of bacterial structural genes that are transcribed together along with their promoter and additional sequences that control their transcription. An inducible operon is one in which transcription is usually not taking place and turned on by the presence of a substance that acts as an inducer. A repressible operon is one in which transcription is taking place but turned off by the presence of a substance that acts as a repressor. There are two types of transcriptional control. Negative control is when regulatory genes encode a repressor that bind to the operator of the operon and inhibits transcription, and positive control is when regulatory genes encode an activator that binds to the DNA sequence and stimulates transcription. Negative inducible system is when regulatory genes encode an active repressor that binds to the operator. This physically blocks the binding of RNA polymerase to the promoter and prohibits transcription. Transcription is turned off if an inducer is present. The inducer binds to the repressor, altering its shape and prohibiting it from binding to the operator, which allows transcription to take place. An example of a negative inducible system is the lac operon in E. coli. The lac operon contains three structural genes, lac Z, lac Y, and lac A. The common promoter for these structural genes is lac P, and the operator is lac O. The regulatory gene lac I transcribes the mRNA that translates to the active repressor that binds to lac O. Without lactose, the repressor binds to the operator, inhibiting transcription. With lactose, some is converted to allolactose, the inducer that binds to the repressor, which releases the repressor from the operator and allowing transcription to occur. It is important to note that transcription is never completely turned off. Low levels of transcription occur in order for lactose to be transported through the cell membrane and for lactose to be converted into allolactose, the inducer. A negative repressible system is when regulatory genes encode an inactive repressor that cannot bind to the operator on its own. Since there is no repressor, RNA polymerase is able to bind to the promoter and transcription takes place. When the co-repressor is present, it binds to the repressor and makes it capable of binding to the operator. So RNA polymerase is not able to bind to the promoter and transcription is inhibited. An example of the negative repressible system is the trip operon in E. coli. The trip operon contains 5 structural genes, trip E, trip D, trip C, trip B, and trip A. These genes encode for enzymes that convert charismate to tryptophan. The regulatory gene trip R encodes for the inactive repressor. When tryptophan levels are low in the cell, RNA polymerase binds to the promoter and transcribes genes into mRNA, which is translated into enzymes that carry out the conversion to create more tryptophan. When tryptophan levels are high, the tryptophan, the co-repressor, binds to the repressor, which alters its shape, allowing it to bind to the operator and inhibit transcription, and no more tryptophan is made. Positive inducible system, regulatory genes encode an inactive activator that cannot bind to the DNA and transcription does not take place. When inducer binds to the activator, it turns into its active form and is able to bind to the DNA and stimulates transcription. In the positive repressible system, regulatory genes encode for an active activator that binds to the DNA and stimulates transcription. In the presence of an inducer which binds to the activator, transcription does not take place because RNA polymerase is not able to bind to the promoter without the activator.